Good afternoon, everyone. My name is Kristen Hagen, and for my UROP project, I have been studying a blood vessel on a chip microfluidic platform for modeling diabetic vasculature and facilitating immune cell interactions for drug screening. 10% of the world population has diabetes, but actually 50% of the deaths caused by diabetes are actually caused by cardiovascular disease that results from this inflammation. This is because diabetes causes cardiovascular changes. Hyperglycemia increases oxidative stress and inhibits nitric oxide production, and this has downstream consequences that eventually lead to overall endothelial dysfunction, inflammation, and atherosclerosis. In vitro cell culture and animal models are currently used to study diabetes-induced changes in the vasculature, but these models are insufficient. So I've been exploring the vessel on a chip system. This is the model that we use to cast our devices. In here, we can seed a collagen hydrogel scaffold and seed cells in the channel. The first aim of the project is to optimize and validate the microfluidic blood vessel on a chip as a platform for replicating healthy vascular function. We seeded endothelial cells into the channel, and we added shear stress to replicate what you would see in the body, and we're left with this three-dimensional channel. The second aim is to recapitulate the characteristics of vascular inflammation and dysfunction that are caused by diabetes. We induce diabetic-like conditions by adding pro-inflammatory cytokines, TNF-alpha and IL-6, as well as a high concentration of glucose. Then we looked for different symptoms of diabetes that you would see in a person on the chip. One example is basement membrane protein deposition. We saw higher expression and deposition of collagen and laminin in diabetic vessels, and this models how the cells, when they're inflamed, kind of deposit extra basement membrane protein as they prepare for remodeling. We saw higher ICAM expression in diabetic vessels, where these surface integrins will tell immune cells where to go. We actually facilitated this immune cell interaction by injecting monocytes into the channel and seeing how they adhere. In a healthy person, your monocytes just flow through your blood vessels. But in a patient with diabetes, they tend to actually adhere and stick to the walls of the channel. This causes them to pass into the intima and form foam cells, which leads to atherosclerosis. And we're able to see this on our chip. Final thing that we looked at is the vessel permeability. When we injected this green dye, we saw that healthy vessels were able to hold on to this dye, but the diabetic vessels were much more leaky, and this is like what you would see in a patient with diabetic retinopathy. Our third aim is to reproduce the cellular responses to diabetes drugs in order to assess the potential for using this model as a tool for drug testing and discovery. We saw reduced ICAM expression after drug treatment, as well as reduced monocyte adhesion. We can conclude that the blood vessel on a chip mimics the behavior of both healthy and diabetic blood vessels in vivo. We showed how this platform can be used to facilitate immune cell interactions and investigate the mechanisms behind endothelial dysfunction. And finally, we saw that reducing the symptoms of diabetes after treatment with drugs shows that this can be a viable tool for pursuing future drug discovery efforts. Moving forward in my project, I will compare measurements to literature and physiologically relevant values. I will actually conduct an experiment to replicate foam cell formation from monocytes on the chip, and I will continue my collaborations using the chip for various applications. This is my amazing team. Thank you so much for your time.